Hello my dear students. In chapter 3 Plant Kingdom, in last session we have discussed in detail about bryophytes, and we have seen that bryophytes, they only grow in damp and humid places, and we have learned the structure as well as vegetative and sexual, asexual, reproductive means, etc. And today we are going to start with the subdivisions of bryophytes. So first they are included as liverworts, and secondly they are included as mosses. Let us start in detail. So first we will discuss about characteristic features of liverworts. Liverworts, they are included as bryophytes, so usually they are found in moist and shady habitats such as banks of rivers or streams or marshy ground, damp soil, bark of a tree, or in deep wood which are full of moisture or humidity. Then if we see the plant body of liverworts, it is thalloid, thallus type, just similar to algae structure. For example, Markentia. The thallus is dorsiventral and closely pressed to the substratum. So flat structure is observable, leafy structure is observable. Then leafy members have tiny leaf-like appendages in two rows on the stem-like structure. And if we see the reproductive mechanism, reproduction occurs asexually, and it takes place through fragmentation of thalli. So it breaks and develops into new, or by formation of special structures known as gemmi, or gemmi are found in the gemma cups. So gemmi are green multicellular asexual buds which develop in small receptacles called gemma cups. You can see in the diagram, which are located on the thallus. So these gemmae become detached from the parent body and germinate to form a new individual. So whenever they get detached, they form a new plant body. So these gemma cups, they are important. Then sexual reproductions, they develop organ, particularly on the thalli, different thalli, and the sporophyte is differentiated into foot, seta and capsule. And after meiosis, spores are produced within the capsule. So these spores germinate to form free living gametophytes like Ternidophytes and Archegoniophore. I will show you in the diagram further. Then we discuss about mosses. Mosses, the predominant stage of life cycle of moss is gametophyte, which consists of two stages. First stage is known as protonema, which develop directly from the spore, and it is a creeping green branched and frequently filamentous stage. And second stage is known as leafy stage, which develop from secondary protonema as a lateral bud. So they consist of upright slender axis bearing spirally arranged leaves, and they are attached to the soil through a multicellular rhizoids, branched rhizoids, very much similar to roots of higher plants. And this stage bears sex organs. Then we talk about the reproductive mechanism. It occurs through vegetative means by fragmentation and budding. Then sexual reproduction, they develop antheridia and archegonia at the apex of leafy shoots, and after fertilization they develop zygote, which develops into a sporophyte, and it consists of foot, seta and capsule. Then these capsules, they contain spores, and spores are formed after meiosis. So the moss have an elaborate mechanism for spore dispersal of spores. Few examples are funaria, polytrichum and sphagnum. As I have explained you earlier that how the bryophytes they appear, their body is of thallus type. So in this diagram, these two, this diagram and this diagram, it is showing the body is thallus, and thallus, they are developing the rhizoids. The rhizoids, they are root like structure, you can see both here, and then the gemma cups, they develop as a reproductive organ. Similarly, the Ternidophyte, it bears antheridium, and Archegoniophore, it will bear archegonium. Archegonium represents female reproductive part and antheridium represents male reproductive part. So this is of thallus type, the body is of thallus type. So here they are known as liverworts like Markensia. And another type of bryophyte, they appear as they are having root, stem and leaf like structure, but exactly true root, true stem and true leaves they are not present, but rather they appear like these structures. So they are considered in Funaria as well as sphagnum. So this is showing Funaria and this is showing sphagnum. Here we can see the rhizoids. So here are the root like structures. Then this is the main axis which is appearable as stem, and this phase is gametophyte. Here gametophyte stage is main stage, and this sporophyte that is dependent upon gametophyte. So sporophyte stage, it bears a seta which is tubular part and it consists of capsule. Similarly here in sphagnum, the archegonial branch, it develops archegonium which is a female reproductive part, and the antheridial branch, it develops male reproductive part, antheridium. Now in this way you are supposed to write in your classwork. So first subdivision of bryophyte will be liverwort, then characteristics, then reproduction, how it takes place, asexual reproduction and sexual reproduction. So asexual takes place by gemme, and sexual reproduction, it takes place by fusion of male and female gametes to produce zygote. So in this way by diagram, with the help of diagram also it is shown. Then next will be showing about mosses. So mosses are another type of bryophytes. Then you are supposed to write down the characteristic features and then reproductive mechanism. Here also vegetative reproduction by fragmentation and budding, and sexual reproduction by developing archegonia and antheridium, and the fusion of gametes and formation of zygote. So in this way you are able to see the alternation of generation. Here in Ternidophytes the main phase is in bryophytes the main phase is gametophyte, and then it is alternated with sporophytic phase. So gametophyte is gamete producing phase and sporophyte is spore producing phase, and they alternate with each other. So alternation of generation we have understood earlier. Now let us start the detail of next higher group of plant which is known as Ternidophytes. Here Ternidophytes mostly they include horsetail and ferns. So ferns, we are familiar with this term, and it is an ornamental plant also. At many houses we develop ferns in our pots. Ternidophytes are mainly used for medicinal purpose also, and they are soil binders also, so that they can protect the soil erosion. And as I have said to you, they are frequently grown as ornamental plants. Ornamental plants are those plants which are developed for beauty, so they are known as ornamental plants. Now if we see the main feature of Ternidophyte, they are the first terrestrial plants to possess true vascular tissue. Now what are vascular tissue? The xylem and phloem are known as vascular tissue. You are familiar with these two terms, xylem and phloem. Main function of xylem is to conduct water and minerals, and main function of phloem is to conduct organic food from one part of the plant to another. So they bear for the first time true xylem and true phloem. So this is one of the main feature of Ternidophyte. We can identify the Ternidophytes like this by observing true xylem and phloem. Then if we see the habit and habitat, Ternidophytes are found in cool and damp shady places, just as algae and bryophytes. Some may flourish well in sandy soil conditions also. Then if we see the main plant body, the Ternidophyte, the main plant body is sporophyte, which is differentiated into true root, true stem and true leaves. So this is a comparative point. We have seen in bryophytes earlier that they were lacking true root, true stem and true leaves, but here for the first time these structures are well developed and well defined. The other structure in pteridophyte, leaves are small, which are known as microphyls, as in Selaginella, or they may be large, which are known as macrophyls, as in ferns. And here if we see the alternation of generation, the main stage, that is sporophytic stage, that means spore producing phase is the main phase, and this sporophyte, it bears sporangia that are subtended by leaf like appendages known as sporophylls. In some cases these sporophylls may form distinct compact structure like strobilus or strobili, it is a plural word, which is also known as cones, like in Selaginella and Equisetum, that I will show you further in the diagram. Next point, we will be discussing about the reproductive mechanism. The reproduction, the sporangia which are produced on the sporophyll leaf, they produces spores with the help of meiosis in spore mother cells, and the spores, they germinate to give rise to a very small but multicellular free living, mostly photosynthetic thallus or thalloid structure, which is known as gametophyte, and it can also be known as prothallus because it is developing. Then the gametophyte, they bear male and female sex organs, which are known as antheridia and archegonia respectively. Antheridia are male reproductive structures and archegonia are female reproductive structures. So these words, they are familiar. We have learned the same concept in bryophytes. Then here, water is required for transfer of anthrazoids. So the male gametes, they are released from the antheridia and they reach up to archegonia, then fusion will take place and the zygote will be formed. The same procedure will be done. Then zygote produces a multicellular, well differentiated sporophyte, which is dominant phase here in pteridophyte. So this is one of the point of difference if we compare bryophyte and pteridophyte. Here in pteridophyte, if we see the alternation of generation, the sporophytic phase is the main phase, and gametophyte, it is dependent upon spore producing phase. In majority of pteridophytes, all the spores are of similar type. So they are known as homosporous. Homo means similar. So when the spores are similar, they are known as homosporous, like in Selagilena and Salvinia. These are other example where two different kind of spores, they are present. One may be larger, so they are known as macrospores, and some may be smaller, they are known as microspores. And here these kind of spores, if they are developed, they are considered as heterosporous. So Selaginella and Salvinia, they are heterosporous, they produce different type of spores. Then if we see the further classification, then pteridophytes, they are divided into 4 classes. Their names are Salopsida, Lycopsida, Phenopsida and Pteropsida. Only the names, they are important to learn. Okay. Diagram, they are showing pteridophytes. This is the Selaginella, and B diagram, it is showing Equisetum. So these two are different type of pteridophytes, and pteridophytes, they bear true root like structure. So here the roots, they are shown. These are the roots which are developed from the main branch, and here also the rhizome, it bears root like structure. Similarly, true stem is present. You can see here the stem, they are observable. This is also the nodes and internodes, they are shown. This is a part of stem. Then the leaves, they appear. Here also the leaves, they are present, and in the same manner it will develop a strobilus. So this particular structure, it develops in one of the branch, and this strobilus, as we have understood earlier, it bears the reproductive structures. So sporophyte, it bears sporangia, and sporangia, they are present on the sporophyll. So sporophyll, it is a very distinct here, it is compact structure and it forms a cone like shape. So this shape, it is very much resembling to the cone. It is a compact structure, and this structure is known as strobilus, in which the reproductive structures they develop. Next, I have shown you that how to write about pteridophytes. Firstly, you will write down the characteristic feature, in which you will explain habit and habitat, then their structures and their examples, they will be included. And diagram is not compulsory to draw, but you are supposed to identify the parts which are present, which are described in detail earlier in your chapter. Then next, I have shown you how to write about detail for related to reproduction, about homosporous, about heterosporous, their examples, and in detail how the prothallus that is formed, how the alternation of generation they are explained. So all these points are important, and always write down all the details point wise, not in a paragraph form. So point wise will help you in gaining the marks. So just develop the habit just to write down the point wise. In earlier sessions also I have told you how to write the points. So first you are explaining habit and habitat, then about related to the structure, then the reproductive mechanism, their types like vegetative, asexual, sexual, and lastly the diagrams, examples. And if the detail of diagrams and examples are given, then you are supposed to write that. So in this way we end today our session here only. Do revise all the topics again and again, it will help you in remembrance. Till then, goodbye students, take care.